By now, we've learned how to name epithelial tissues. Now we're stepping into glands and how to name them. And so first up, we're going to talk quickly about an exocrine gland and its basic structure. Exocrine glands comprised of a stroma. The stroma is a fibrous outer capsule. If I had to give you an analogy of what the fibrous outer capsule would be like, it'd be like taking an orange and looking at the orange, peeling the orange off and looking at the white covering between the orange peel and the actual orange. Sometimes it seems to be thick, sometimes it's thin, but it's kind of a layer between the peel of the orange and the actual fruit that you want. And that would be our fibrous outer capsule. And then that orange gets divided up into wedges by that same fibrous outer capsule coming down and kind of dividing it out into those wedges. That would be considered the septa, extensions of the capsule that divide that orange into wedges, or in this case, divide this gland into lobes or lobules. Okay? So on our picture, this is the stroma on the outside, and then how this comes down into it and divides it into lobes or lobules, that'd be the septa. Okay? When we get down into the deeper portions, we have what is called the parenchyma. And here they've labeled the parenchyma, and then we're going to blow it up so we can take a look at it. The parenchyma, which is this, blown up, is broken down into two parts, the duct and the acinus. The acinus is where we actually have the simple cuboidal cells that synthesize the secretion. Okay? And they're simple cuboidal. The other portion is the duct. The duct is essentially epithelial tube, doesn't really matter what type of cells they are, that line this tube that allow the whatever was secreted to be conducted to wherever specifically we want it to go. Okay, and that's the duct, the epithelial tube that drains them. Okay, and that's the simple structure of an exocrine gland, pretty basic. Now, we can name these exocrine glands, and it's very similar to naming epithelial tissues. First, we look at the number of branches. Okay, if it has one branch, or excuse me, like one duct that has no branches, that's simple. If it has a duct that has many branches, or more, not many, more than one branch, then that's compound. So here's an example of a simple exocrine gland. You see one branch, or excuse me, one duct with no branches. And over here, these would be compound. We see a gland with multiple branches, more than one, compound. So simple means it has no branches. Compound means it has branches, more than one. Okay. Then, when we named epithelial tissue, we looked at the cells on the apical surface. Here, we're going to look at the shape of the gland itself. So, if it's simple, it's simple. If it's compound, then we look at the branches. So here's a couple of branch ducts. We're going to look at these branches. We can call this a compound acinar gland or a compound tubuloacinar gland. And remember, the acinus is where the secretion was made, so it's where the secretory cells were found. And they've tried to indicate those in purple here. Okay? So we look at this, and here's the secretory cells, secretory cells, secretory cells. It's almost like they're at just the ends of the ducts. They're at the cul-de-sac here. Okay? Down here we have the secretory cells, but they also go up the street. Not only just in the cul-de-sac, but up the street. So this acinar gland, the secretory cells form a dilated sac instead of a tube. So they're found down here in the sac or the cul-de-sac, like what you think of in a subdivision. The tubuloacinar means the secretory cells are not only in the sinus, but in the tube that would normally be conducting. Now it also has some of those secretory cells there. So this would be having those not just in the cul-de-sac in the neighborhood, but the major street through the neighborhood, leading to even more major street intersection. So secretory cells found both in the tube and in the acinus. So another way we can classify these glands is by their secretion. Okay? So we have an exocrine gland, we look at it, we look at the type of secretion it makes. If it's a thin, watery secretion, like tears or digestive juice or sweat, then that's a serous secretion. Serous is describing that thin, watery secretion. If it's a thicker secretion, one that involves generally mucin, mucin's a glycoprotein that absorbs water to form a thicker substance, the mucus, then they're considered to be mucus glands. So serous glands, thin secretion, mucus glands, thick secretion. Sometimes these glands can be mixed. Salivary glands fall into this category where they're both serous and mucus, oftentimes being secreted together. So we just think of them as salivary glands all at once. But if you've ever noticed, maybe if you're doing public speaking and your sympathetic nervous system is kicked in, that's actually innervating the mucus glands. So you start to produce more mucus secretion, which absorbs the water in your mouth, and you start to get cotton mouth. You feel all nervous. Okay. Now, parasympathetic innervation innervates more of the serous secretions, and that's when you smell food or you're going somewhere for Thanksgiving, you start to get that mouth watering feel. So that's categorizing these glands based on their type of secretion. We can also categorize them quickly on how they secrete, their method of secretion. And what such way we could do that is we call it a holocrine gland. In a holocrine gland, it's like a volcano erupting. These cells that contain the products actually break up, disintegrate, and release the products that way. So not only is the product being released, but also the cell fragments and dead cells will come up with it, kind of like a volcano. Not only is there lava, magma, there's also dirt and whatnot, earth. Okay, so secretory cells disintegrate. They deliver their accumulated products in some of the cell fragments. Oftentimes the oil-producing or sebaceous glands associated with hair follicles will do this same thing. And then opposite or different than a holocrine gland is a merocrine gland, of which there's another specialized merocrine gland called an apocrine gland. And these glands use excitosis. And you have to remember what excitosis is, maybe from high school biology. But it's the process of which it develops the products inside the cell, and they're contained within a vesicle. And that vesicle moves through the cell, fuses with the plasma membrane, and then releases its products, in this case into a duct. When it does this, it maintains the integrity of the cell membrane, and so the cell isn't damaged or destroyed. So it just releases the products. Okay? So excitosis is used by a merocrine gland to release its substance. Examples of this: lacrimal, gastric glands, pancreatic glands, all use these. All where we don't want the cells to fall apart. Okay? Where we need them to continue doing what they were doing. Now, an apocrine gland is a fancy merocrine gland, and generally the difference is not only does it release the products, but they also get some cell cytoplasm within that vesicle, and that's released with it. Now, with that, there's oftentimes some amino acids that get released in that cell from the cell cytoplasm, and the thing about that is that bacteria tend to like these secretions. They come, they eat those amino acids, and then they, in their breakdown, they release off some gases of their own, and we know that as body odor. Okay? Apocrine sweat glands are found in the axillary and inguinal regions, meaning they're found in the armpit and in the groin. And these are the ones that are most specifically responsible for body odor.